Chapter 7.2 is all about catabolism, which is breaking down molecules. So that's the first part of metabolism. They call it the microbial buffet, right? This is essentially like microbes eating and digesting their food. So you take complex food molecules and break them down into what are called catabolites. So you're converting complex molecules into smaller things and in the process releasing energy, which needs to be stored in some intermediates because we're going to then take that energy and build things with that. So we're going to talk about two molecules that are energy carriers. This molecule called ATP, adenosine triphosphate, which we've talked about, and another one called NADH, which is going to help transfer energy from energy-producing reactions and transfer that energy to energy-spending reactions that are going to build new molecules. So again, catabolism, I can't say this enough, is the breakdown of complex, big organic food molecules into smaller components. Oftentimes, the simplest process is using carbohydrates, sugars, starch, things like this. You can use lipids or fats, and proteins can be broken down as well. There's more steps in this process, though. In the process of breaking these down, energy is going to be released, and a lot of that is going to be transferred into ATP. Some of that is going to go to NADH as well. Smaller biomolecules, amino acids, right? When you break down a protein, you release amino acids. When you break down fats, you release fatty acids. Something long like starch, when you break it down, you release simpler sugars like glucose. Those can be produced in the process. And what you can eat or what you can catabolize is dependent on what enzymes you have. So here's an interesting side note. Starch is just a bunch of glucose molecules stuck together in a particular arrangement. So you can see this part here is always sticking upwards in starch. So you can eat starch, right? It's the sugar that's in potatoes. They're so energy dense because they have these long starch molecules in there. Cellulose is almost identical. It's made up of the same glucose molecules, but they're arranged in an alternating pattern. So cellulose is a main component of plant cell walls. That's what makes plants stand up tall and woody plants firm. You cannot digest cellulose. So if you eat a bunch of celery, you really don't get much nutrients from it. Most of it gets excreted. That's the dietary fiber part, right? That's because you have enzymes that can break down the starch bonds here, but they're so precise that they will not break down the almost identical bonds in cellulose here. So humans can't break down cellulose, but you can break down starch. Now there's microbes that can break down cellulose and use it for energy. A lot of biofuel companies are very interested in these microbes because they can take plant biomass, parts that we can't digest and eat, and break it down into fuels and things like that. Also, in the guts of ruminants like cows sit there and chew on grass and things like that, but it's actually the bacteria inside of their gut that can break down these cellulose bonds and release the energy that cows then can use to build biomass, which is hamburgers, right? So that's just one way you can use food sources. And one of the themes in metabolism is that we have a bunch of kind of different inputs, right? You could take carbohydrates, like these long polysaccharides. They're going to feed into the pathway. You could also take fats, and they feed in slightly differently, but they end up in the same pathway. Some microbes can use lignin from wood or other what are called aromatic compounds, things that are even found in petroleum and stuff like that. They can feed them into a very similar pathway here. Amino acids, peptides, proteins, they can feed into this pathway as well. And you get different amounts of energy out of these different processes, but they all feed into this central pathway. So this is what we're going to focus on here, is pretty much the flow of carbohydrates into this metabolism pathway. And this is going to flow into what's called oxidative phosphorylation, potentially. There's a couple of different ways this can go. These parts, we have this process called glycolysis that's very universal. You do this and bacteria do this. We have the breakdown of pyruvate, which is an output of glycolysis. They have two options. You could go to fermentation, or you go into what's called the TCA cycle. This is also called the Krebs cycle or the citric acid cycle. That's spinning around and around, breaking down this molecule. But we take a big molecule and we break it down through these different steps here. Each of these arrows here is controlled by an enzyme that does the regulation of that reaction there. Having this one central pathway is very, very efficient evolutionarily. The reactions that occur in here are oxidation reduction reactions or redox reactions. They always occur together because an oxidation reaction is the substrate, whatever it is, donates an electron to some other substrate which accepts electrons. That's reduction. So one thing gives up some electrons, sends it to another one, which gets reduced. So oxidized, reduced. I'll show you some drawings of this. I would like you to know what these terms mean because what's happening here is these electrons are energy, and that energy is being transferred from one molecule to another. So in the case of our catabolism here, biomolecules donate electrons for oxidation, and they're going to give them to these electron carriers, NAD, FAD, and ATP, which accept electrons. That's called reduction. So we're breaking down our food and giving the high energy electrons to these intermediates. This energy is then going to be used to build new molecules in anabolism. So the way to remember oxidation reduction is Leo the lion says grr. So loss of electrons, oxidation, Leo, gain of electrons, reduction, grr, right? So here you can see Leo says grr. Here you can see Leo says grr. All right. Hopefully you can memorize that. We will have some examples of where this is happening now. You're going to take the energy that's produced from breaking down food molecules, and you got to store it somewhere. In the case of ATP, we start with what's called ADP, adenosine diphosphate. So it's got two phosphates on it. Remember, each of these phosphates has bonds in it that are stored energy. And if we take another phosphate and use some of the energy that's produced from those reactions, we can hook that third phosphate onto the molecule. That's storing the energy in a chemical bond. So we go from a low energy state here, we stick that phosphate on, using the energy, we have higher energy, potential energy here. Then this ATP can wait until it's called upon. When we need to power another reaction, we can chop these phosphates off, and that will power other reactions that can build other molecules. The same thing happens with this molecule called NAD+, and its reduced form NADH. So we have something that oxidizes, loses an electron, and this, what it's called a nicotinamide ring, NAD+, it's going to accept 2 electrons, and in the process, some hydrogens come with it. So the electrons are donated to this molecule, forming NADH, which is the high energy state of it. And this can transport those electrons and give them to a process called the electron transport chain. This electron transport chain, it's also called the electron transport system, is the energy that makes this hydrogen ion gradient, or the proton motive force. We will see this. We talked a bit about it previously, but we'll see it again. So this is taking electrons from the food molecules, storing it temporarily, and then helping use it to make this gradient. This gradient is going to produce lots of ATP, which is a more stable energy storage molecule than this NADH. Okay, so we've talked about energy. Now we've talked about breaking things down and how we transfer the energy around. It's through high energy electrons. So the catabolic pathways, they organize the breakdown of large molecules into smaller pieces, and that happens in little steps, step by step by step, all controlled by enzymes. And in the process, they're releasing energy that's going to be stored in smaller molecules like ATP and NADH. ATP stores that energy in bonds. You take a diphosphate, add another phosphate, it becomes triphosphate. Hydrolysis, or breaking of those bonds, is then going to yield energy when you need it. NADH, on the other hand, stores a pair of electrons that will go to the electron transport chain in oxidative phosphorylation, which we'll talk about in a little while. All right, that's 7.2.